Pleasure to be here and be able to speak to you today. So today I'm going to be talking about some of the work that I've been doing over the last 5 years. And so I will be talking during approximately 25 minutes. So yeah, feel free to, maybe if you have a burning question, then maybe jump in, but then maybe perhaps, after the 25 minutes, there will also be an opportunity to talk. Okay. So basically, I'm going to give you basically a background of where I'm coming from, what is my expertise, and what is my research focus. And then I will be telling you about the different methods that we can use and we have developed in our group to map chemical changes in DNA. And then in particularly, I'm going to talk to you today about a project on mapping a type of DNA damage known as abasic sites, and then the bioinformatics method that I developed to be able to do that. And then a potential collaboration of some of the work that is happening there in Utrecht, and then where maybe a mapping abasic sites might be of interest. And then if we have some time at the end, I will briefly introduce you to some other projects that I've been working on as highlights, and then we can later on perhaps maybe chat about that by email. Okay, so I have a background in chemistry, and I'm originally from Spain, but during my undergraduate, I spent quite a lot of time at different universities in the US, and I was in South Korea for a while as well. And then, as Patrick was saying, and then I went to the EBI, and then I started on a project looking at, so just like there are algorithms to compare the sequences of proteins or the 3D structures of proteins, we wanted to develop an algorithm to be able to compare reactions, so biochemical reactions, but also synthetic reactions with an aim to be able to search the literature as search databases for similar reactions that would help us to maybe design new reactions in collaboration with chemists, or design enzymes for applications such as, biodegradable products or maybe applications in agriculture, etc. So, and then I co-developed that tool, and then I've also applied it to study the evolution of enzymes. Okay, so just for you to have an idea of what is my PhD background. But then towards the end of my PhD, I wanted to learn more about all the technologies that were developed to map mutations and chemical modifications in DNA. So I joined the group at the chemistry department at the University of Cambridge, and also the Cancer Research UK to learn that as well. So the Balasubramanian group is a very multidisciplinary group, just like many of the groups there in Utrecht. We are really a combination between chemists and biologists, but also bioinformaticians. And then we work together in different projects, and in this case, to map different chemical modifications in DNA. So these are our two sites. We're based in the chemistry department, but also at the Cancer Research UK, which is perhaps a bit like the Princess Maxima Institute in Utrecht or the NKI in Amsterdam. And then so our research focus is we study epigenetics in DNA. In particular, we go beyond the process of mutation, but also we look into processes of chemical modification. So, for example, you have a cytosine in DNA that gets methylated, or it gets oxidated, or other chemical process. This is the work I'm going to show you today at the seminar. And then later on in the year, hopefully I can meet you in person, and I'm going to be telling you more about some other area of work that I've been doing is in mapping these secondary structures in DNA known as G quadruplexes. Okay, so as you know, DNA and the encoding of information in DNA is just not about ACGT, but it's also about all of these chemical processes happening, such as modifications in DNA, DNA methylation, histone modifications, also the structuring of DNA around nucleosomes, and then the structuring of nucleosomes and DNA chromatin, and the packing of DNA in genome structures within the nucleus. Okay. So particularly the questions that we are trying to address in my research is whether these chemical modifications exist. We're trying to find out where are they located and how are they produced. What's their function, their role in development, and also in disease. And then more from a structural point of view, we are interested in finding out if they alter the structure of DNA, if they modify chromatin, or if they affect transcription factor binding, if they have any effect on polymerase activity or gene expression. Okay, so here I'm showing you three modified bases. So on the very left-hand side, this is a T modification called 5-HMU, which is not known at the moment where it's present in mammals. There is some preliminary evidence in mouse, mass spectrometry evidence that it might be present, but funnily enough, this is a modification that is very common in parasite organisms. So just like in mammals and higher eukaryotes, we have that mainly DNA epigenetics happens in cytosine. In parasites, however, all of these epigenetics tend to happen in thymine, right? So in T, which is very interesting. So on the middle, I'm showing you 5-methylcytosine, which is very abundant in mammals and bacteria as well. And on the right-hand side, I'm showing you a very common modification known in adenine, okay, N6-methylase. It's currently being investigated whether in mammals, so there is some contradictory evidence in the literature of whether that's the case. It's definitely very abundant in RNA, but its role in DNA hasn't really been nailed down yet. Okay, but still, it's very common as well in bacteria and nematodes, so worms. Okay, so I'm sure many of you know. So this is the hydrogen bonding pattern of DNA. You have G in front of C and A in front of T. And these are the hotspots where these chemical modifications take place. So starting clockwise, you have that position so in the major group of cytosine where it's heavily modified. Then just like in cytosine, at the same position, in thymine, you tend to have a lot of modifications as well. So in adenine, you have that N6-methylase modification that I was mentioning happening here in the nitrogen. And then in guanine, you have modifications happening here on the 7th and on the 8th position of the guanine, okay? So how these modifications appear? So I'm showing you a lot of chemistry here, but just the idea here is very simple. There are enzymatic reactions happening in the cell that are responsible for these chemical modifications. So for example, you have the methyltransferase introducing a methyl group here, okay? And then you have oxidation enzymes that oxidize that methyl group down to hydroxy and carboxy, for example. So that's if you look at the cytosine pathways. If you look at the thymine pathways at the bottom here, you also have these oxidation processes, just like you see in the cytosine, or even more exotic reactions. For example, the addition of a glucose molecule, which is a sugar, to DNA, which is quite interesting to link DNA and sugars. Okay. But then you also have these excision enzymes that are responsible for detecting the modification, cleaving the modification, generating an abasic site, which is shown here in the middle, called AP. And then later on, there is another enzyme machineries known as the base excision repairs that are responsible for fixing these abasic sites back to the original base. Okay, so you can see that these abasic sites, they play a central role in these mechanisms of excision and repair. And they also not only play a role in enzymatic processes, but they are also intermediates in processes of oxidative DNA damage, where during the repair of this oxidative damage, they also go through abasic sites. Okay, so just briefly, I'm going to give you a quick overview of the methods that are known to map chemical changes in DNA. So there are two main methods. The first one is known as mass spectrometry. And then the idea here is you take DNA, you digest it with enzymes into their individual nucleotides. And then using chromatography and mass spectrometry, you can map the mass of these individual nucleotides into a chromatogram shown here. And then if you have a standard chromatogram where you're basically, you're trying to find out what you're looking for, so the modification that you're looking for, you can essentially compare your chromatogram for your DNA with your standard chromatogram and be able to tell which modifications are there. Okay, this is a very sensitive technique, and then it allows you to determine the global levels of modification. And then it tends to be very high. It's very high sensitive, and it's the first method that people use to discover new and unknown modifications. So one of the limitations of mass spectrometry, it doesn't tell you where in the genome the modification is present. Okay, so for that, there is sequencing methods. So the way it works is that, again, you fragment DNA into small DNA fragments, and then using a pull-down strategy with a specific chemistry or an antibody, you can enrich for the reads that contain the modification that you're interested in, and then doing high-throughput sequencing, and then you can determine the position in the genome where this modification is present. So it allows you to do a genome-wide map of the modification, and then also it allows you to determine, in some cases, in which nucleotide exactly the modification is present. Okay, so just a brief overview of the strategies, if you like, by resolution. So there are single nucleotide methods. For example, some of you might have heard about bisulfite sequencing. So these are chemical conversion methods where you use a chemical to change a base into another base. So for example, here cytosine goes into uracil, which is then in the sequencing machine read as T-stymine. However, some of the modifications upon the chemical treatment, they are unchanged, and therefore by looking at differences, then you can tell whether a given modification is there or not. So that's one family of methods. Another one is by taking advantage of the fact that when you have a modification, you can modify it with a specific chemistry, and then that will allow the DNA polymerase to stop at that position. And then by looking at this asymmetric distribution of aligned reads, that will allow you to tell if there is a modification present or not. Now, more recently, there are technologies that rely on single molecule real time. So some of you might be familiar with PacBio sequencing. And that also allows you to detect modified bases, or nanopore sequencing, whereby, you know, if you have a DNA that is modified, then that given modification will generate a differential of potential across the nanopore that will also allow you to detect that. So other low resolution methods rely on using chemical enrichment or antibodies that you can use them for pulling down the reads that contain the modification. Okay, so just in the last 10 minutes or so, I'm just gonna briefly tell you about this project on mapping abasic sites. So this is work in collaboration with Jane and Fumi. Fumi is now in Japan, actually. She's at the Riken Institute in Japan. And they are both chemists, and we've been developing this method together over the last 3 years. So abasic sites are a form of DNA damage. And as you know, DNA damage plays a fundamental role in as a hallmark both in cancer and in aging. In particular, the role of DNA damage in cancer goes through this genome instability and mutation hallmark. And similarly, in aging, you have that a component of aging has to do with a genome instability, but also alterations into the epigenetics, so a modified basis in DNA, but also histone modifications. So, so one form of DNA damage is induced, as I mentioned earlier, due to reactive oxygen species, for example, in guanine, okay? And then there are the mechanisms how these are repaired, but fundamentally, abasic sites are an intermediate of all of these mechanisms of repair, of excision and repair. So in a way, the cell, when there is damage happening in the cell has a dilemma. It has to decide on whether to repair the modification or whether to leave the modification as it is, and perhaps that will lead into mutations, and then later on perhaps cell senescence, and well, perhaps the development of a tumor in cancer, or the development of neurodegenerative diseases, for example, Alzheimer's. Okay. So these are the two known methods to map abasic sites. So what I'm going to be talking to you today about is our method, which is here shown on the right, which allows us to detect abasic sites and single nucleotide base resolution. So I'm just going to show you the method in more detail. So an abasic site, as you can see, is just a position in the genome where the base is missing, so you don't have a nucleotide. Okay, so you can see here in the middle. So you have your phosphate backbone, but then you have bases, but then it's just missing. So we know from mass spectrometry evidence that there is about 10,000 of these happening per cell and per day. And then, as I was mentioning in the previous slides, there are products of DNA damage, and then key intermediates in this mechanism of excision and repair. The way we detect them are by, we've developed a chemistry called the HIPS chemistry, where we can tag abasic sites specifically, and then we can also degrade the DNA fragments all the way to the position where the abasic site is present. And then with the tag, then we can enrich using streptavidin pull downs. And then by sequencing the position of the read one, if you do parent sequencing, the position of read one is determinant of where the abasic site is located. Okay. So this relies on being able to tag abasic sites specifically, And here I'm showing you the equilibrium for those of you that has a background in chemistry. This is really an equilibrium between the closed form of the ribose ring and the open form of the ribose ring, when you have an aldehyde and then you can exploit that aldehyde to be able to tag abasic sites and do pull down. Bioinformatically, what you're come around is a situation like this. So this is, I'm trying to represent you an alignment here. So you have a treatment library where you see these pile-ups of reads, from the forward, aligning to the forward or aligning to the reverse. And then you're looking for positions in the genome like that, right? Where you clearly see a change into the pile-up, okay, of reads. So you're looking at positions, right? So here I'm doing some counting, for example, right? So we got, 1, 2 reads starting at that position, but suddenly you have a position in the thymine that you have 9 reads starting, right? Or, if you look at it from the reverse strand point of view, similar situation, right? You have in the treatment libraries, you have at that position 12 reads that start, but then in the control, you have only 2 reads, right? So you can think already, how then we can detect those differences, all right? So I'm just giving you a brief overview of the bioinformatic pipeline to detect these sites. So obviously we take the sequencing reads and then we quality check them and trim them. And then we do alignments using BWA. And then at the end of the day, we end up with our alignments, and then we calculate alignment counts. And then we filter. So obviously we have counts for each position in the genome. So we do quite a lot of filtering on just positions that have significant amount of counts. And then we normalize the counts by library size, okay? So then we do some modeling. So we use modeling that is very commonly used in RNA-seq or ChIP-seq methods, which they rely on these negative binomial distributions that are appropriate for count data. And then given that we have multiple replicates for the treatment and the control, then we have to estimate the variance. And so then we fit GLMs, so generalized linear models, to the treatment and the counts, just like you would do in RNA-seq. And then we score the individual sites. So under the null hypothesis that there is no difference between the treatment and the control. And then finally, so we do multiple testing corrections, and then we do volcano plots and the thresholding, okay? So and then you end up with volcano plots like this one, right? Where you threshold at different levels of significance, and then you extract. So essentially here, these are individual and single nucleotide positions in the genome. And then depending on the level of astringency that you want to put, then you can select a more relaxed or more stringent threshold. Okay. And finally, what it takes most of the work then is when we are collaborating with the chemists and the biologists to really find out how we can make sense out of this. So we profile the genome, the sequence context, we do motifs, we look at associations with gene expression data sets, etc. Okay, so this is an example of how the data looks like. So this is from one of our libraries, not in the human genome, but in a parasite genome known as Trypanosoma. Okay, so here you can see on the top, there is a region of the genome that is highly enriched. So the blue is the treatment library and the green is the control library. And then if you zoom into that region that is enriched, then you see these sort of stacks, right? These, if you like, stair-like positions where then you can detect them with our method. And then, for example, these are from 3 positions in the forward, 2 positions in the reverse that they have been called to be abasic sites. Then later on, then we can do this, we can look at the positions, so which positions are more abundant. So we, in this particular libraries that I was showing you, the modification, it was really abundant in thymines, okay? Given that it's coming from a parasite library, which they have epigenetic happening in these, then this is something that we were expecting, that all the abasic sites are coming from these modifications. And then we're also just beginning to apply it to human libraries. So here I'm showing you some examples that we're working on at the moment. We've been knocking down AP1, which is one of the key enzymes for repair, the base excision repair in the genome. And then we are also trying to see if there is any particular regions in the genome that are abundant in abasic sites. So these were quite preliminary. So we're seeing that there seems to be quite high abundance here in showing you in yellow in functional regions, in promoters, UTRs, exons, and not very enriched in introns and intergenic regions. Okay, this is something that is something that we're working. It's work in progress. And then, so one of the things, just to finalize this section, one of the things that we think is interesting is in this project that is coming up from actually some of the labs in Utrecht, the lab of Ruben van Boxtel and Hans Klever, and also Edwin Copenhagen, where some of you might be familiar with this study. They've been looking at a chemical produced by E. coli. It's called colibactin, okay? And then within E. coli, as you know, is present within our gut. And then they've been able to identify in this publication just from a few months ago that the colibactin molecule generated by E. coli might be responsible for the development of colorectal cancer in patients. And then they've done a really nice study on investigating then the mutations profiles. They've been able to find out that colibactin acts in adenines, okay, and they seem to be sort of anchoring in 2 points at the same time, so in the forward and the reverse strand, perhaps. But there is still quite a lot of work to be able to find out what is the actual structural intermediate, if it goes through an abasic site mechanism, or perhaps through another mechanism. And we think that perhaps our technique to map abasic sites might be able to give some insights on whether the colibactin mechanisms go through abasic sites. Okay, so just a thought of potential application of our technology. Just finally, last slide. This is some of the work that we've been working on as well. I didn't have time today, but happy to catch up with you if you're interested later on. So we've been mapping cytosine modifications in patients that suffer from glioblastoma, which is a type of brain cancer. And then we have some preliminary data where we've been able to show that the tumor component, they tend to have disappearance, so a loss of this type of oxidative modification. This observation has also been made in patients that suffer from liver cancer from different groups. So we were doing further investigation on whether this 5-HMC could potentially be used as a biomarker for brain cancer, which is one of the most aggressive types of cancer out there. Another interesting piece of work that we've been doing is we're trying to explore whether this modified base called 4-methylcytosine actually reacts with the lysines of, for example, nucleosomes and histone molecules on a shift-based mechanism. And whether, so then we were generating these maps of 4-methylcytosine, and then we're comparing it against the position for nucleosomes to see whether there's any correlation. And then we also have some biophysical data showing that that interaction is happening in vitro. Finally, another project that is very exciting, and then we're just starting that at the moment, is on mapping thymine modifications in parasites, just following up from our work in mapping abasic sites. And then the interesting here is that, you know, parasites are very, you know, parasitic agents that are very aggressive in some parts of Africa, especially in Central Africa. And then we want to understand more about how we can inhibit the growth of these parasites. So by knowing more about the biology of T modifications in these organisms, then we might have some ideas of how to develop new therapeutics. Okay, I'm happy to talk to you about that later on if interested. Okay, so just to finalize, yeah, just say thanks to the group. It's a very interesting place to work. So if you're thinking about, you know, maybe for your PhD or postdoc are interested in genome, feel free to drop a line. It's a very inspiring place to work. And then just obviously to thank the entire Balasubramanian group, and then also our funders. And so yeah, happy. Thanks for your attention as well, and thanks for Patrick and Adrian for inviting me. I'm happy to take any questions.